So this is going to be translation. If you remember, we had a genetic code which was copied, okay, which was opened up and copied. So this was our DNA, which has got adenine, thymine, cytosine, and guanine joined up in a sequence. And these are these little codons. Okay, that could be A, T, C, 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 A, G, C. We opened it up. So it was opened up like this. And both sides then lined up, and we made an RNA copy. Okay, and the RNA, instead of T, so T has been replaced with U. Okay, so the RNA copy there would have been this piece of RNA here is called messenger RNA, and if that was an A, then this side would have been a T, an A, a G, a G, a G, a G, a T, a C, and a G. So this mRNA would have been on this side here. That would have been an A. Okay, that would have been a T, but now it's a U, and this would have been a C, and again this would have been a C, 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 and this would have been A, C, G. Okay, and it's this was called transcription. This is like you photocopying the main library, and you take this piece of mRNA, okay, that's your M or messenger RNA, okay, and with its code AUC, CCC, ACG, and that, it would be longer than that. This has got 3 words in it, 3 codons, if you like. There are lots of possible ones. And what happens is there are things called tRNAs, and they look a little bit like this. Okay, there's things called tRNAs, look a little bit like this, and they have corresponding codons on them like this. Okay, that one might be UGC or GGG or GAU, like that. These tRNAs, they will land on here in the right sequence. Each tRNA has what's called an amino acid on it. An amino acid. An amino acid. And these amino acids, when they come down next to each other like this, they land, the amino acids, okay, so these are landing down. These land on this in the right sequence like that. They join. These amino acids, there are over 20 different types, and they join together, okay, and this goes off and forms a long chain which folds into different shapes, and that forms a protein. Okay, now there are about, as I said, about 20 different types of amino acid. Each amino acid is coded for by a different one of these. So one of these codons, that one might say a particular type of amino acid, that will say a different type of amino acid, that will say a different one. So you end up with lots of different amino acids all put together in a particular sequence. Okay, so the gene here that's been transcribed from the DNA into the RNA here, then you translate the language of genes, okay, A, C, G, U, T, etc. You translate that into the language of proteins. And if you put different amino acids together in a different sequence, it forms a chain which folds on itself, and it forms different shaped proteins. And those different shaped proteins, remember when we draw enzymes, they have shapes, and all enzymes are proteins, and enzymes are what drive our body. Okay, so enzymes, and they have a shape, and that shape is determined by this sequence on the mRNA, which is in turn a copy of the sequence that's on the DNA. So that is how proteins are formed. Okay, and obviously if there's any changes to these letters here, it will change these letters here, and it could mean the wrong amino acid being put in the wrong sequence in this protein, which means maybe this protein is the wrong shape, which means maybe it doesn't do its job. Okay, and a change in the DNA is called a mutation, and this is how mutations can affect proteins, and proteins affect how our bodies actually work. One thing I have forgotten is that all of this process here takes place inside something, okay? Inside an object here called a ribosome. A ribosome, okay? So you get the mRNA going in, the ribosome sits on it like that, comes through, and the proteins come out the other side like this, okay? And that these then form into the protein. So all of this process of translation from mRNA using tRNA to form proteins takes place inside the ribosome.